Hey Aria. I had to jump in and go live because I saw your message. I wanted to know what it was. You did your speech and it went amazing. Oh, well done. I know you were a little bit worried about that, weren't you? Aria, do you mind me asking what it was on? Your speech. I'm glad it went well. You've got parents' evening tonight as well, right? Hopefully that will go well as well. And then you've had a brilliant day. I'm sure it will. I'm sure you'll have a good parents' evening. Oh, well done. Good. Proud of you because you were nervous. You were nervous yesterday, I remember in the lesson. Oh no, now we're talking about boys. Go on then, you can tell me. You can tell me what they asked you. Hi, natural scientist, how are you? Welcome. Welcome to today's live. The plan is really to just do some questions with you guys if you're up for it. I've got some paper one questions, so it's going to be suitable for year 12 or year 13 to do some revision. Oh, Aria, what did you say? Yeah, so if you're year 12 or year 13, stick around. We're just going to spend like half an hour, 40 minutes. We're going to do some random questions. I think I've taken these questions from 2022 A level paper one. So we've got a mixture of stuff. Hey, Chinwendu. Aria, are you sticking around for this? I'm just thinking, Aria, have we done this yet with year 12? I know Chinwendu, you're in year 13. Natural scientists, remind me what year you are in. But yeah, stick around. We're going to do this question first. Describe viral replication. So Chinwendu, you should be able to do this. Aria, you could have a go, maybe. I can't remember whether we've done this, Aria. You've done it in school. Okay. Now this just says viral replication in general. But I suppose if you're AQA, the one that we actually learn about is HIV. So you could answer it in terms of HIV specifically, or you could just answer it in general for a virus. So, anyone want to start us off, or shall I do this one? I've kind of just dived straight in with quite a difficult question. I do realise that. But yeah, obviously with these lives, if you want to really test yourself, you should be trying to answer the questions with me, putting your suggestions into the chat. As you guys will know from my lessons, I always use bullet points literally for every single question. So for this, I'd be looking for 3 bullet points, but I'd probably do 4 or 5 just because we know what biology mark schemes are like. So I'd try and say as much as possible. Aria's talking about viruses there. Yeah, they are not living things, are they? They are not made of cells. They don't carry out metabolic reactions. They don't respire, for example. They can't replicate unless they're in a host cell. Good knowledge there. But for this question, we have to describe specifically how they replicate. So for example, we could say things like, oh, I've used the wrong there. Meant to be the other there. We could say their attachment proteins bind to receptors, which are also made out of proteins, on host cells. Now, if this was about HIV specifically, do we know which kind of cell they actually bind to? Yes, Chinwendu's just preempted my question. If this was about HIV, then HIV has attachment proteins that bind to receptors on T helper cells. They're called CD4 receptors on T helper cells. Now, you could have said that and you would still get the mark, but you don't have to say that because it just says viral replication in general, okay? So their attachment proteins bind to receptor cells on host, receptors, sorry, on host cells. And then their nucleic acid enters the host cell. Or if we're thinking about HIV, it enters the T helper cell. Guys, what is the nucleic acid in HIV? What type of nucleic acid do they have as their main like genetic material? because I've just said nucleic acid, because again, I'm keeping it vague, right? But yes, if you were specifically talking about HIV, you could say the RNA enters the host cell. If you're talking about other viruses, some other viruses have DNA, so you could say their DNA enters the host cell. It doesn't matter here because it hasn't specified which specific virus this is. So their nucleic acid enters the host cell. Guys, I'm going to test you now. If it was HIV, which does have RNA, as some of you have told me, what other step could we include to potentially pick up a mark? What has to happen to that RNA in order for it to be incorporated into the host cell's DNA? Yes, Aria, loving this from you. Yeah, so if we have gone down the HIV route, we could get a mark for saying RNA is converted to DNA using reverse transcriptase, which is the enzyme that literally does the reverse of transcription. So instead of making RNA from DNA, it's making DNA from RNA. Now, we can actually get a mark for saying that, but we might not include it because we might not be specifically thinking about HIV. But if we are thinking HIV and we've gone down the route of RNA, we could have included that and picked up a third mark. Other marking points available, we can say viral proteins are then produced. And we can also say new viruses are then assembled and released from the cell. Or they will bud off from the cell. That's obviously going to cause cell lysis. So the host cell is going to be destroyed, it's going to lyse, it's going to burst as these new viruses are released from the cell. So they were the marking points for that question, allowing students to talk about just viruses in general or specifically HIV, which has RNA converted to DNA. But yeah, I mean, how do we feel about this topic? This topic, by the way, is not on OCR. I don't think at all. They don't do viruses, they don't do viral replication, but it's on AQA. And because it was a new topic that AQA added to the spec, there have been quite a few questions on viral structure, viral replication, HIV, AIDS, all that kind of stuff. Yeah, Aria, is that why you're super engaged today? You're feeling super happy? That's good. Yeah, this wasn't bad. Okay, good, because some students do find this really, really hard, I'll be honest. Right, let's see what the next question is. Oh, we've got a table. We love a table. Don't get many easy questions like this on biology, but we can get 2 marks. We've got some features, and all we've got to do is tick to say whether it's talking about mitosis, which is obviously cell division that makes body cells, or binary fission. Thanks, Aria. Loving having you here. You are giving me all the right answers. If we're replicating linear DNA, that would be mitosis, right? Because DNA is linear in a eukaryotic cell where we get linear chromosomes. Circular DNA would be binary fission because prokaryotic cells have circular DNA. They have a big loop of circular DNA that we call the nucleoid, and they also have plasmids, which are circular. What are you saying for 3, Aria? I think you're saying mitosis and binary fission. Yeah, both. Both of them produce 2 daughter cells. Produces 4 daughter cells. That's neither of these. Which type of cell division produces 4 daughter cells? Can anyone answer that? Yeah, you're right, natural scientists. So binary fission happens in prokaryotes, mitosis happens in eukaryotes. Should we fill that in? There we go. Good. Hi, Michael. Yeah, natural scientists, you're right. 4 daughter cells, that would be meiosis, which is the type of cell division that occurs to make gametes or sex cells. That makes 4 daughter cells, and they would be haploid because they would have single chromosomes. So that's like a bit of a red herring. You should not tick anything for 4 daughter cells. But if you get this column correct, you get 1 mark. And if you get this column correct, you get a 2nd mark. So I think that's a super, super easy question. Let's see what else we've got. Maybe a harder one. Ooh, this looks harder. Who's done meiosis? Meiosis is kind of towards the end of the year, I would say, in year 12. Obviously, year 13, Chinwendu, you've done it. But this is about meiosis, and it's about non-disjunction, which makes it even harder. Yeah, Aria, you haven't done it. Well, you can kind of remember back to GCSE though, right? Meiosis makes 4 daughter cells, and it has 2 divisions, not just 1. So you can kind of think back to GCSE and you might still be able to do it. But you won't know what non-disjunction means. Non-disjunction basically means the chromosomes have not separated properly. And that's really all we need to know to answer the question, because it tells us non-disjunction has occurred in the chromosome labeled N, which is this one here. So it looks to me like the first division has happened correctly. In this first division, the first division of meiosis, because we know it has 2, in the first division, we separate the homologous pairs. And that has happened correctly because we've separated the first pair and we've separated the second pair. But in the second meiotic division, or the second division of meiosis, we should be separating the sister chromatids. But we are told that that has not happened correctly for chromosome N. So on this side, it's happened correctly. So we'd separate the 2 sister chromatids of the smaller chromosome, and we'd separate the 2 sister chromatids of the larger chromosome. That would get us 1 mark. That's happened correctly. But on this side, it hasn't happened correctly for chromosome N. So all we need to do, the little one has separated correctly, but chromosome N hasn't. Okay, so we could draw that. Does that make sense if you've not done this before? We're saying chromosome N has not separated as it should. So those 2 sister chromatids have not separated into those 2 daughter cells. So they're still going to be stuck together. They're still going to be attached by their centromere. So we could draw it like that. Or, guys, if you drew it like this, that would be absolutely fine as well. Doesn't matter, as long as you show that those 2 sister chromatids have not separated. That's what non-disjunction is. And here, it's happened in the second meiotic division. So the pairs were separated correctly, but then the sister chromatids were not separated correctly. The result of this, by the way, this gamete, I'm not sure if this is a sperm or an egg. Don't think it tells us. But this gamete has got 2 copies now of that chromosome. It's got identical copies of this chromosome. Yeah. So that if that gamete is used in fertilization, the offspring will have an extra copy of that chromosome. Okay. And it can cause, I mean, like, this is what causes Down syndrome, right? An extra copy of a chromosome. So it's quite interesting. Okay. Who's still with me? I'm a bit reluctant to do this question. Has anyone seen this one before? Have I had lunch yet? I've not had lunch. I'm really bad though, Aria. Like, I'll quite often not have lunch because I'll just stay in here and I lose track of time and I keep working and I'll skip lunch and then I'll end up having just like a snack later on. But Aria, if you're hungry, go and get lunch. Go and get lunch. There's no problem. Or you can eat and watch on your phone. Right. Who wants to have a go at this maths question? This was on the 2022 paper, I'm pretty sure it was. I know that I gave this question to students in my class when I still worked in a college, and you'd be surprised, loads of them couldn't do it. Even students that do A-level maths. Don't know what it is about maths in biology. You just seem to hate it. But yeah, I'm going to do this with you because actually it's not that hard. But let's go through it. Doctors are studying babies born with a mutation. Kind of, yeah, should read all the information, but I do tend to skip through it, which is bad. They determine the mother's age at the time of childbirth and whether the non-disjunction happened in the first meiotic division or the second meiotic division. A student concluded there were more mothers over the age, that means greater than 37, with MM2 errors than with MM1 errors. Use the graph and calculations to show why this is not correct. Right, so we're only looking at mothers over the age of 37. And I think this is where some of my students got it wrong. They included mothers that were 37, but it just says over 37. So we only want to look at these bars, okay, 38 plus. Let's have a look at the number with MM2 errors first. So if we look at the key, the grey bars are MM1 and the white bars are MM2. Yeah, we need to do some calculations though to prove it, Aria. So if we look at the white bars for MM2, we've got 0.29 and we've got 0.07. So 0.29 add 0.07. But look at the total numbers as well, because there were different sample sizes in these two groups. Should have been able to do that in my head, really. So 0.36 had MM2 errors, but look at the total number, 230. So we do 230 times 0.36, okay, to get the total number. So 230 times 0.36, and that gives us 82.8 for the MM2. I think what's difficult is the total numbers were different, and the graph has given us a frequency. So 0.36 of 230 had MM2 errors. It's almost like 36% of 230 had MM2 errors. Good, James. Now let's do exactly the same for MM1. And let me read these off accurately. So we've got 0.22. I've got 0.04 and 0.01. And I am going to do it on my calculator. Don't judge me. You've got one, so why not just check? So many students, obviously you could do it in your head and then make a silly error and then lose the mark. So 0.27, that's kind of like saying 27% of the mothers had an MM1 error, but there were 730 in that group. So I need to do 730 times 0.27 to get the total number of MM2 errors. And what do I get? 197.1. Oh, I'll write it down here. So what I've just proven is that actually there are more MM1 errors. So MM2, there was 82.8, and MM1, there was 197.1. Yeah, exactly. And if you look at the question, Aria, it says, using figure 2 and suitable calculations, show why this conclusion is not valid. It is not valid that more mothers over the age of 37 had MM2 errors than MM1. That is not correct, because looking at the calculations, actually more mothers had MM1 errors than MM2 over the age of 37. Does that make sense? Because look at the total numbers. The numbers in the groups were different. The mothers with MM1, there was 730. The mothers with MM2, there were only 230. So even though the bars, it looks like MM2 is higher frequency, it was a higher frequency in a smaller group of mothers. So actually, there's more MM1 errors than MM2. Now the marks, just so you know, because James, you would have got both of them, the marks were literally for these numbers. You didn't actually have to do any writing. You just had to use calculations to show that actually the MM1 errors were higher than the MM2. So if you got 197 or 197.1 and you got 83 or 82.8 and you could see those numbers in your answer, you would have got 2 marks. Okay, good. Thanks for sticking with me. I know a lot of students will just like skip through the maths questions, shy away from the maths questions, but remember the maths in A-level biology is no harder than GCSE higher maths. What's difficult is that you quite often get a load of text and a graph or a table, or you get all of those things and you have to figure out what maths you're supposed to do. That's what's hard. The maths itself, I mean, look, it's literally just multiplication and addition and then comparing. But it's just figuring out, isn't it? And I think what threw people is over the age of 37. So we're only looking at the last 3 groups of mothers. Natural scientist, is astrobiology hard? Oh, I wouldn't know because I've never studied it. I don't even know if that was like a course available for me when I went to uni. I mean, it sounds like it would be really interesting, but I'm guessing probably quite difficult. Do you know what kind of things you study in it, natural scientist? Have you kind of looked into the courses? Because that's the best thing to do, isn't it? Find out exactly like what topics are covered. And if it sounds like stuff you're going to be interested in, then even if it's hard, you'll probably make a good go of it. If you enjoy something, you're always going to work harder at it, aren't you? Is it a course in America? I think they probably do that in the UK though now as well. It includes most of the natural sciences. Yeah, there'll be aspects, I should think, from lots of different branches of science. Because I'm guessing it's like about life in space, is it? Am I being really dense? Life in space, that would be my understanding. So you're going to have to look at like all different aspects. Let's do a couple more questions. I've gone for an easier one at the start here and then a slightly harder one. Right, good, Aria. You didn't need to say 3D structure. You just needed to say the second part of your answer. Usually when we define tertiary structure, we say 3D structure, held together by hydrogen ionic disulfide bonds. And then when we say quaternary, we just use the second part of your answer. Although there's no harm in saying what you've said. Like, you are right. The quaternary structure is going to be 3D as well, right? But all you need to say is the quaternary structure consists of more than one. It's as easy as that. Or you could say 2 or more. Just another question for you, if you're still with me. How many polypeptides does hemoglobin have? How many polypeptides does hemoglobin have? How many polypeptides does hemoglobin have? Aria, we'll get on to the next question in a second. I don't think you would get a mark there because you need to be a bit more specific. Yes, 4. 4 polypeptide chains in hemoglobin, 4. And then 4 heme groups, which is why each hemoglobin molecule can carry or transport 4 molecules of oxygen. What about an antibody? Have you done that yet, Aria? How many polypeptides does an antibody have? Antibodies obviously made by plasma cells, which are a type of B lymphocytes. No, not done that yet. That's 4 as well, weirdly. Another one with 4. It's got 2 longer polypeptides, which we call heavy chains, and 2 shorter polypeptides, which we call light chains. But obviously, different proteins have different numbers. It's just that those 2 examples on your spec both happen to have 4. Right. Weird question then, because we are always taught that enzymes are specific. But this question is saying, how could 2 enzymes with different amino acid sequences catalyze the same reaction? So they've got a different order of amino acids, but what do we think the active site must be like? It's not linked to the induced fit model, although that's quite a good thought, because we do know with the induced fit model, the active site does change shape slightly to mold itself around the substrate. But think about the whole enzyme. The amino acid sequence is different, but it's the active site specifically, isn't it, that binds to the substrate. So what might we be thinking about the active site of these two enzymes? Because that's the bit that matters. That's the bit that needs to be complementary to the substrate and bind to the substrate. I'm going to show you how I would answer this question. There's a couple of different ways, but you could suggest the active site region may have the same sequence of amino acids, because the active site is just part of the enzyme, right? The whole enzyme is like a big globular protein with loads of amino acids. It might be that the sequence of amino acids is different elsewhere, but in the bit that forms the active site, the actual sequence of amino acids there is the same, and therefore the bond formation in that region is the same. What else could you suggest for marking point 1? The active site region may have the same, or you could say the active site may have the same tertiary structure. So it might be that the rest of the tertiary structure is different, but that bit, do you see what I'm saying? That bit of the enzyme actually has the same shape or the same tertiary structure. Yeah, no, Michael, it is tricky. I'm going to be honest with you. This is a hard question. I can totally understand why you wouldn't have said that. The other thing you could say, this is still marking point 1, is the two enzymes may bind to different parts of the same substrate. So it might be that one enzyme's active site is complementary to that side of the substrate, and the other enzyme's active site is complementary to that side of the substrate. Do you see what I mean? So maybe the tertiary structure of the enzyme is different, maybe the active sites are different, but maybe the active sites are complementary to different parts of the substrate. So they can actually both bind to the same substrate. And that's another suggestion. The two enzymes, I'll put active sites in there, I think, may bind to different parts of the same substrate. Marking point 2 is a standard marking point. And what I say to all my students is whenever you get an enzyme question, try to say somewhere in your answer, enzyme substrate complex. And you can actually get marking point 2 for saying, so they can both form enzyme substrate complexes with the same substrate, obviously. Let's have a look, James. The differences in the enzymes allow them to regulate the same. Oh, do you mean isozymes or enzymes, James? I'm not quite sure what you've said there. But if you've just said the differences in the enzymes allow them to regulate the same reaction, that would not be worth any marks, unfortunately. What do you mean by isozymes? What do you mean by isozymes? I'll go get some lunch after, Aria, I promise. Yeah, so even if you had no idea for marking point 1 and you just kind of panicked, if you just stuck down, well, they both form enzyme substrate complexes with the same substrate, at least you would get marking point 2. But yeah, either their active site is complementary to a different bit of the substrate, or maybe their active site actually has the same tertiary structure and the same shape, because maybe the order of amino acids is actually the same in that bit of the enzyme, and the tertiary structure of that bit is the same, is kind of what we're saying. James, I'm lost. I don't know what you mean by isozymes. It's not a term that I've used before in A-level biology, unless you've got advanced knowledge here, maybe. But yeah, that was the mark scheme. So there's a few alternatives to marking point 1, and then marking point 2 is like standard. Let me just give you a few tips, because I've got yeah, probably a chemistry term. Yeah, too long since I did A-level chemistry. Multiple forms of enzymes. Well, that would make sense, wouldn't it? Because it's like an isomer. But you're kind of told in the question, aren't you, that you have got 2 different enzymes. You're kind of told that in the question. So I don't think they would be able to give you marks. What I say to my students, if you get any enzyme question, I always say to them, think about using your key terms. So I'll just list them. Have I missed any? I basically just say, look, you should have these words in your answers. So you should be talking about the tertiary structure of the enzyme, the active site, the fact that it's complementary to the substrate, and the fact that it can form enzyme substrate complexes. That's what I say. And quite often, if you miss those words out, you don't get the marks because you've not used the key biological language in your answer. A-level chemistry is so hard. I agree, Aria. Definitely the one I found the hardest. Yeah, I'd say it was definitely the one I found the hardest. I'm trying to think back. To be honest, it's like a blur for me, A-level chemistry. All I remember, I might have said this to you before, I remember talking a lot about carbon, drawing lots of structures with carbon, and doing lots of titrations. I honestly felt like every time I walked into the lab, there was, what's it called? Is it called a burette? Is it called a burette? I'm looking really thick now. We had like burettes. Is it a burette? The big glass things. We had them on the desks, and like every lesson, we were doing a titration. I remember, that's all I remember, is doing titrations all the time. But yeah, it is hard. Hi, Ifa. Yeah, you think of burette. That's the term that came to my head, but then it's been so long that I was questioning it. Yeah, and you had to like neutralize an acid with an alkali or something, doing a titration. That's literally all I remember. Right, guys, anything else? Are there any last questions? You think biology is harder? Yeah, I mean, Michael, actually, student. I mean, I probably shouldn't say this to you because you're obviously doing biology, but students do worse in biology than chemistry on average. And I think that's a combination of there's more content to learn in biology, like way more content. The mark schemes are way more specific. And you've still got a lot of application and practical questions and maths questions as well in biology, like you do in chemistry. So yeah, students actually tend to do better on average in chemistry than biology. James has confirmed it is a burette. Nice. Nice. Yeah, so guys, I mean, don't worry. You're obviously all dedicated students because you're here spending your lunchtime with me. So I'm sure you're all going to put the effort in required to get good grades in biology, but it is going to be a lot of work, right? Because main reason is just because the mark schemes are so specific. Yeah, we can have a bit of a chat, Aria. I like to do some past paper questions because otherwise I feel like you're not getting that much out of it. So I like to do a bit of revision. Um, so I usually pick a few different questions to go through. You've given up on maths questions in biology. Oh, don't say that. Remember, Michael, it's not going to be harder than higher tier GCSE maths. So that should reassure you and give you confidence. So what my advice would be is if you get a maths question, just read the information carefully. Check, usually in biology math, there's some issue with units. So I would like underline the units that they've used in the question because there's usually some kind of unit conversion in there. But yeah, read the question carefully, underline the key information, check that you know what you're doing because the maths itself is actually going to be straightforward. It's just doing the right maths. Okay, yes, right. I'll go then, guys. I'll grab some food. Enjoy your lunch. Thank you for joining me today. And I'll see you next week. Not sure what time it's going to be next week. Might be Thursday lunch again. But if you keep an eye out on social media, we will let you know. And yeah, I'll pick some more questions, some different topics that we can revise together. Yes, I'll see you on Monday in lesson, Aria. Have a lovely rest of your week, guys. Thanks for being here. James, what's your quick question? Put it into the chat and I will try and answer it if I can. What is your quick question? What food you're going to have? Do you know what? Our cupboards are pretty bare. Haven't got a lot in, Aria. More questions on transport in plants. Okay, cool. So we're talking like transpiration, cohesion tension theory, translocation. Nice, Aria. You're doing a past paper question and you're stuck. James, give me a bit of info on the question and maybe I could slot it into next week's live. Do you know what the question, can you type the question in? Do you know what paper the question was on or what number the question was? Because I could try and find it. Is there any histology involved? Are you doing OCR or AQA? There's a bit more on OCR because you do a bit more about like the histology of the pancreas and the liver. But there's not much really on AQA, no. Yeah, no, not really on AQA. I mean, we obviously learn about microscopes and we do look at cells and tissues under the microscope. You might have to do a biological drawing, but no, not really. Yeah, not really on AQA. You're right, Aria. There's a bit, I think for OCR, you do look at like the islets of Langerhans in the pancreas, the endocrine and the exocrine tissue. You look at the liver a little bit, the hepatocytes in the liver, but not for AQA. James is still typing his question. Okay, I'll hang fire. I'll hang for a minute. Oh, right, James. It's written it down. Let me get my pen, James. 4 marker. I love it. I love how specific you've been. What topic is it, by the way? So it's June 2023, paper 1, A level, not AS. Question 3.5. Nice. I'm going to have a look now and see what it is. James, can you come back next week? Indian food, yeah, I love a curry. I mean, I don't really make it myself, but I get a takeaway, Aria. And Indian's probably my favourite takeaway. Okay, James, come back next week if you can. My favourite curry would be a lamb bhuna. You know what's making me laugh though? I've watched a lot of Gavin and Stacey over the holiday because I watched the Christmas special and then I went back and watched like the first 2 series and it makes me think of I don't know if you've seen Gavin and Stacey, you know, when Smithy orders his curry and he's like lamb bhuna, prawn bhuna, and he orders like 4 or 5 different curries. Aria, you need to watch Gavin and Stacey. It's a classic. Gavin and Stacey, they're all available on iPlayer. How have you not heard of it? It's James Corden, Ruth Jones. You don't have time. Oh, you can each episode's like 25 minutes. Watch one and you'll love it. Have you watched House MD? No, what's that about? Let me write this down because I need some good TV recommendations. I've just finished watching Missing You on Netflix, which I loved. I loved Black Doves. House MD. What is it about though? Because I don't like anything cheesy. Yeah, watch Gavin and Stacey. How is it? I like things about like, you know, like thrillers, like murder, gripping kind of thriller mystery type things where there's a lot of drama. Is it like that? You're like my kids, Aria. I'm like, I go in, they're watching YouTube. I'm like, why aren't you watching proper TV? They literally just watch YouTube. I don't understand it. It's a medical drama. Okay, I might like that. I'm writing all this down. Right, I'll watch the trailer. Thanks for the tip, guys. Nice one. Think of anything else that I might like, let me know. Right, I'm gonna go and see what food I can find. Mac and cheese. I had macaroni and cheese for tea on Tuesday. It was a ready meal though. I didn't make it myself. Right, I'll let you go. Right, bye guys. Thanks for being with me today. See you next time. And I'll look that question up now, James. Bye.